The polymerase chain reaction, or PCR, is a way to amplify DNA as well as isolate the DNA of a gene. And the goal of a PCR reaction is, let's say we have the genome, and in this genome is a gene shown in blue, and what we would like to do is to selectively amplify the DNA or copy the DNA of this gene so that we have lots of copies of this gene at the end of the PCR reaction. So let's say that we would like to amplify this blue region of DNA, and this blue region would be called the template. And the template is usually buried in or is part of a bigger piece of DNA, such as the genome. And it so happens that we know some of the DNA sequence at either end of the template. So here we know that the template starts with ATTGGA and it ends as CGTAAT. For simplicity's sake, we are only showing 6 base pairs, but usually you would want to know the sequence for at least 20 base pairs. And here's our 5 prime, 3 prime, 3 prime, and 5 prime. So in order to carry out a PCR reaction, the first thing you need, of course, is the template DNA. The second thing that's needed are primers, and the primers are going to be used to carry out a DNA synthesis or DNA replication reaction. And so we would have 2 primers, one that's complementary to the left of the sequence, and so the primer also has a 5 prime and a 3 prime end, and it has a hydroxyl group on the third carbon of the A, so that the when DNA synthesis begins, the first base pair can form a phosphodiester bond with by carrying out a condensation reaction with this hydroxyl, and the other primer is complementary to the sequence on the right of the template that we would like to amplify. And so it would be ATT, ACG, ending in a hydroxyl on the third carbon on the G. Once again, so that you can add new nucleotides after that. Then, of course, we need, if you want to do DNA synthesis, we need a DNA polymerase. And usually the DNA polymerase that's used is from the extremophile Thermus aquaticus, which lives, or which was discovered in the hot springs at Yellowstone National Park. And this DNA polymerase from Thermus aquaticus, called Taq polymerase, is thermostable and can withstand and maintain its activity at the high temperatures used in the PCR reaction. And it works much better than DNA polymerases from E. coli. And of course, if we are doing a DNA synthesis, we need deoxy nucleoside triphosphates. So DATP, DTTP, DCTP, and DGTP that will be polymerized by DNA polymerase during the synthesis reaction. There are other components as well. For example, you need a source of magnesium ions necessary for the function of DNA polymerase. The first step in a polymerase chain reaction is to heat the reaction mixture to 95 degrees Celsius. And what this does is it denatures the DNA. The high temperature causes the DNA molecules to vibrate and move around so much that the hydrogen bonds are broken and the two strands of DNA separate. And then in the second step of PCR, the reaction mixture is cooled to a temperature between 55 Celsius and 70 degrees Celsius. Cooling the reaction mixture down now causes hydrogen bonds to form again, but instead of hydrogen bonds forming between the template DNA molecules, hydrogen bonds form instead between the primer and the single-stranded DNA molecules. So we will have the primers making hydrogen bonds with the template DNA, and this is known as annealing. The primers to the template DNA. In the third step, the reaction mixture is heated to 72 degrees Celsius, which is the optimal temperature for the Taq DNA polymerase to carry out DNA synthesis. And when you have DNA synthesis, you will basically extend from the primer, the 3' end of the primer, and make the daughter strand for both the template strands that we started out with. And therefore, now we have where we started with 2 strands of 1 double-stranded DNA molecule. Now we have 4 double-stranded DNA molecules And so we have doubled in these 3 steps by denaturing at 95, then annealing at a lower temperature between 55 and 70, and the third step, which is DNA synthesis, also called extension, we have doubled the number of DNA molecules. And altogether, all these steps, this is called one cycle. Next, during a polymerase chain reaction, this cycle is repeated. So once we have carried out another round of denaturation plus annealing, this is what our DNA molecules are going to look like. We will have now 4 strands, and the primers would be annealed to all 4 strands, so that when we do extension, we end up with a total of 4 DNA molecules, of which 2 are just the region of the DNA that we want to amplify. And in the third cycle, the amount of DNA doubles once again, and we have 4 molecules, 4 DNA molecules that are the template that we are amplifying. And therefore one could make a plot of PCR cycle on the x-axis. So let's say this is C, and on the y-axis we could plot the amount of template DNA, or target DNA, and you could use different units to measure this. You could use the number of DNA molecules, or the moles, or you could use picograms, nanograms, and so on. And so let's say I started out with X amount of DNA that I prepared my reaction mixture with X amount of DNA. Then after the first cycle, I will have 2x the amount of DNA, and after the second cycle, I'll have 4x the amount of DNA, and then I will have 8x, or 2 cube x the amount of DNA, and then I will have, after the fourth cycle, 2 to the power 4x the amount of DNA. And this shows that we have exponential amplification. You double the amount of DNA in each cycle, and that's why it's called a chain reaction. And we can say that the amount of DNA Y after C cycles is the initial amount of DNA times 2 to the power C, where C is the number of PCR cycles. Next, let's look at an example. During PCR amplification, we have 512 picomoles of DNA after 10 cycles. What was the initial starting amount? And we can approach this in two ways. One is to simply write down that equation where we have y is equal to x times 2 to the power c. Now here c is equal to 10 and y has been given to us as 512. Therefore 512 is equal to x times 2 to the power 10, and 2 to the power 10 is 1024, and we can divide both sides by 1024 to solve for x, and we get x is half picomole. And the other way one could do it was simply half the amount, 512 10 times, and we know that 2 to the power 9 is 512, so halving it 9 times will get us to 1 picomole, and if we halve it once more we will end up with a half picomole.